I'm Ramona Brito, and I'm a curator at UniProt, the Universal Protein Resource. I'm joining you from the Welcome Genome campus today, and my colleague Emily, who is our proteomics expert curator, will be joining us from Cambridge. And she will be ready to answer any questions if you have them during the course of the webinar. Also, if there are any questions on proteomics in particular, you should direct this to her. So let's get started. So the UniProt Knowledgebase is prepared by a team of curators and developers who are in 3 different sites, here at the EMBL-EBI, which is on the Genome campus, at the Protein Information Resource, which is in Georgetown University in the US, and the Swiss Institute of Bioinformatics in Geneva. So today, I will be talking in particular about the Proteomes portal, which you can see on the main homepage. And I will run you through three parts of the proteomes data we have in the database. So initially, I'll start with an overview of what proteomes are, where this data comes from, so we understand how we bring this into the database. And I will talk a little bit about how the data in sequence databases, nucleotide sequence databases in particular, and UniProt has been growing in the last few years exponentially, and what we are doing about it. And then I will go on to show you some examples to which I hope will help you identify the most relevant set of protein records for your work. So I have chosen some frequently asked questions, in particular 3 questions asked by 3 different users, to which I hope will be helpful. And there will also be a small section on COVID-19 proteomes, and this will be followed by Q&A. So to start with, what is a proteome? A proteome, as we define it, is the set of proteins thought to be expressed by an organism's genome. So in the case of the human proteome, for example, the genome is known to have around 20 to 25,000 genes. And the human transcriptome is considerably more complex because of things such as alternative splicing and editing of mRNA. You can have up to an order of magnitude more complexity in the transcriptome compared to the genome. So you can have around 100,000 transcripts. And as we know, these transcripts undergo post-translational modifications. So the variety of proteins that you see is even more complex. So you can have more than 1 million proteins, for example. So the main thing to note here is the increase in complexity. So what we have done in UniProt is like this. So if you look at the human proteome in UniProt, you will see that it has 75,000 proteins. So where does this come from? So, so we have 2 different views of the proteome. One is the sum total of all the proteins that make up this proteome, and the other is the gene-centric view of the proteome. So as we know, there is slightly more than 20,000 genes, and we have made an attempt to group together the proteins that originate from a single gene. So you can either look at all of the different proteins available, or you can download 1 protein sequence per gene. So we do this by grouping together the similar genes and then choosing 1 canonical sequence for each group. So the other thing to note here is that we have a very large number of reviewed entries, and this simply means that a curator an expert curator has looked into this and added information, has annotated each of these records with additional information. Right, so I want to go a little bit into where this data comes from. So the data we see in UniProt, where does it come from? So a little bit on the technical side, to understand where these sequences come from, we can think of genome sequencing and genome assembly as solving a jigsaw puzzle. So initially, everything is assembled, and then you have to necessarily chop it up into pieces in order to be able to sequence the genome. And so then you read the genome, these small little fragments, you reassemble them back, put the pieces back together, and once you have large enough fragments, such as contiguous sequences or scaffolds larger than that, then you're able to use gene prediction software, gene prediction algorithms, which can come in and annotate the genome as a whole. So this is where we are able to predict which regions of the genome actually code for a protein sequence. So once we have this information, we're able to translate those sequences and bring them into UniProt as protein sequences. So another view of this is like this. So we have whole genome sequencing, which is done by independent research groups. They're also done by large sequencing consortia. And all of this is submitted into one of the nucleotide sequence databases. So the major ones are NCBI, GenBank, ENA, which is EMBL-EBI, and the DDBJ, the DNA database of Japan. Together, this is called the International Nucleotide Sequence Database Collaboration. So any genome sequence that is submitted to one of these databases is available to be brought into UniProt. So what we do is we translate the nucleotides, we bring them in, but we don't bring everything in. So I just wanted to make the point that we do have filters in place to exclude certain genomes from coming in as proteomes. So the first filter is to exclude anything that is anomalous. And I'll define this as I go on. So if a genome is anomalous, then the sequence is not brought in. Otherwise, it comes into the Proteomes database. So this, I've shown the UniProt archive, which is the archive of all of the sequences that come in from any one of these databases. And then the second level of, the second filtering that we do is for redundant proteomes. Meaning if a proteome is highly redundant to another proteome, then we don't bring it in. So let's go into why we do this. Right, so the first level of filter is for anomalous proteomes. This is pretty straightforward because proteomes can be excluded because they are anomalous, and that can fall into one of several categories. And largely we follow the criteria used by RefSeq at the NCBI. We do have some differences, but I won't highlight them here. So once we have eliminated or excluded the anomalous proteomes, we go to the second level of exclusion, which is redundant proteomes. So proteomes may be excluded because they're highly redundant to other proteomes. So we have a procedure in place, an in-house procedure, that compares each incoming proteome with every other proteome in its species group or in its set of closely related proteomes to see if a particular proteome is largely the same or not. So I've taken the example of one species group which had 7 proteomes, and this procedure identified 5 of these proteomes to be redundant to 2 of these proteomes. So each of these is a different strain of the same bacterium. And so once all of the pairwise comparisons are done, then we can get rid of all of the orange ones, and that leaves us with only the green ones. So if you want to, so what was the impact of this? So first of all, why did we do this? Because the nucleotide sequences were growing exponentially, and therefore the impact on UniProtKB was the same. So at the end of 2014, there was an exponential growth. We reached around 90 million sequences. So it was time to do something. So we employed this redundancy minimization procedure, and I just wanted to show you the small visualization to see what the impact was. So each node here corresponds to a proteome, and the size of each cluster is the number of proteomes available for a species. So this is a species with a large number of proteomes. So after minimization, the sizes were largely reduced. So you have a more granular figure on the right, but the impact on the database as a whole was dramatic. So in April 2015, we implemented this procedure for the first time, and around 50 million sequences were deprecated overnight. So if you want to read more about this, you can look at the paper that was published a year afterwards. And what happened after that? So UniProtKB reduced in size. This was 2015, and the line in orange is the INSDC, which is all of the nucleotide sequence databases. This kept growing as usual. And as you can see, UniPark, which is all of the unique sequences in UniProt, as well as Tremble, have been growing since, but at a much more manageable rate. Right, so this is just a slide to show you the dramatic drop in the number of proteomes for certain species. As you can imagine, highly redundant species were largely reduced in number. So E. coli, for example, had 1,900 proteomes, and we were left with 65 non-redundant proteomes at the end of the procedure. All right, so let's shift gears a little bit and go to the current web interface. This is release 2020-05, which is the current release. Just wanted to mention this COVID-19 portal that we have here if you're interested in these sequences in particular, but I won't go into that right now. So if you click the Proteomes portal, you're confronted with all of the proteomes available. So you can see here that we have nearly 300,000 proteomes as of today, spanning all the different superkingdom, bacteria, archea, eukaryota, and viruses. So it is nearly impossible to navigate this and to find your proteome of interest. So what do you do? So in order to explain what we would do in a particular case, I've taken the example of an actual user query, which I hope will help you understand how to find the most relevant set of protein records for your work. So Manisha Aswal is a student at the University of Delhi, and she wanted to compare two different species of bacterium. Yersinia pseudotuberculosis and Mycobacterium tuberculosis. They are quite different, but they have a common pathogenic trait. So she and her colleagues were interested in comparing these different proteomes from different genera, and hoping to identify some common traits, some common genetic markers that could explain why they had a common pathogenic trait. So where should she start? So let's have a look. First, you start with the taxonomy. So I would suggest if you have a question like this, you go straight to the taxonomy database, not the Proteomes database. So drop down taxonomy and type in your species of interest, and you find you get many different hits. But so So how do you identify which one? So I would suggest going to any one of these and navigating your way. So I clicked on the first one, and you're able to find your way. You can navigate up or down, taxonomy navigation. So you can go up until you find your species of interest. So in this case, this is the species level tax ID, under which there are 18 different nodes, which correspond to different strains of the same bacterium, for example. So the interesting thing here is that 66 proteomes were available, if you see on the filters on the left. So I clicked on that, or actually, I will go back one slide and show you something. If you click on all lower taxonomy nodes, you will get even more than 66 proteomes, because it will include not just this species, but every other subspecies. So on the whole, there were 75 proteomes, but this included non-excluded and non-redundant proteomes. So we talked about the anomalous proteomes and the redundant proteomes. Do we want to include this in the analysis? I don't think so. So the first thing would be to click this. So you eliminate all of the anomalous and all of the redundant proteomes. So we do that first. So if you eliminate the anomalous, you're left with 74. If you eliminate the non-redundant, you're left with just 11 proteomes. So all the rest are redundant proteomes. So the next level of selection is this. When you look at this, so now you have 11 proteomes, and you can exclude lower taxonomic ranks if you don't want them. So that leaves us with 8 proteomes. And you will see that there is a vast difference in protein count between these different strains. So what's happening here? So I want to introduce these two columns, BUSCO and CPD, both of which are proteome quality measures we introduced last year to help users identify the best proteomes for their work. So if you look at these 8 proteomes, you can see immediately that 4 of them have a lot of missing genes. In fact, they're nearly almost missing all of the genes. And CPD is an in-house algorithm which tells us simply if a particular proteome is standard, meaning it looks like other proteomes in its species group among its neighbors, or if it's close to standard, or if it's an outlier altogether. So as you can imagine, the ones with missing proteomes are outliers, and the good complete proteomes are standard, usually. Right, so that's what I wanted to point out. And then, so what I would do in this case is to select the 4 proteomes that are very complete, and then go and download these genomes. So this is what she did, and she went on to do the same thing for Mycobacterium tuberculosis, and selected 4 species of Yersinia, 3 species of Mycobacterium, and by doing this analysis, they were able to identify 7 proteins that may play a role in the common pathogenic trait that these 2 different bacteria exhibit. So it's a very nice study. It was published earlier this year. So if anybody's curious about what these proteins are, you can go and have a look. Right, so the next question is also a typical question that I get from time to time from people who are looking for a particular genome that they know was sequenced by their group or somebody else, but they can't find it in UniProt. So sometimes they ask nicely, other times they don't. And so I went and looked at the European Nucleotide Archive, which is basically the INSDC, and it was missing. So if you look in NCBI, this record, this master record was available, but there were no protein coding sequences, meaning the genome was submitted, but the genome was not annotated. So where did this information go? Because clearly the genome was annotated when they published the paper. So in the end, we found that this information was submitted to a specific database for spinach. So, and then it was not submitted anywhere else. So we told them we were very sorry, we couldn't include this in UniProt. So this happens from time to time, and I went and looked at the spinach genome in UniProt under taxonomy, and I found one proteome, but it's not the same proteome, it's from a different group. Right, okay, so the next thing to address is programmatic access. So some users would like to access a set of genomes programmatically, maybe as part of a pipeline. So they would like to set up a pipeline that automatically picks up a set of genomes and updates them as and when updates become available. So for this, I would suggest using not able to move. Right, the Proteomes API, which you can find at this URL. So as you see on the left panel, you have a proteome section, which lets you get proteomes by UPID. UPID is the unique proteome identifier. So if you have a set of UPIDs already, you can go here and you can put them in, and you'll find what is necessary to plug into your pipeline. But if you don't, then I would suggest, again, going back to taxonomy and identifying which proteomes you actually want. So taxonomy is always a good place to start. Right, so the last question came from Saugata Roy at the University of Leicester about COVID-19 proteomes. Do we have COVID-19 proteomes? And if so, where can I find them? So the obvious answer would be the COVID-19 portal, but the COVID-19 portal is different. It basically has COVID-19 proteins. If you want to look at proteomes coming from whole genomes, then you go to the Proteomes portal as usual. So again, a good place to start is taxonomy if you don't know which organism you're looking at. So fortunately, if you type in COVID in taxonomy, it pulls out the right taxon, 2019 and COV. And as you can see there, it says one proteome, but let's have a look. So if I go into the taxon page, the page for this taxon, I find that there are actually 92 proteomes. Right, if I follow the link to the 92 proteomes, then I can see that there is one reference proteome and 91 other proteomes. So this proteome has been chosen by the curators at UniProt to serve as the reference proteome. So this is the one you should pick if you want to choose. So if I click on the reference proteome, I find that it has 14 proteins, typical for a virus like this. And as you see here on the left panel, 13 of them are reviewed, the SwissProt entries, meaning curators have already annotated them with information. So curators are working on this and keeping the data as up to date as possible. And so this is the set of proteins, one long polyprotein and all of the other proteins. All right, so that's it from me. And Emily and I will take questions if you have any. So there is a question about, okay, what can they do in the case of spinach proteome to get it included into UniProt? It's a question from Sadiq Zahari. can they simply upload their data into INSDC? Yes, that's what they should do. Sometimes if it's a popular proteome and many users want it, we have requested the authors to submit it, but often we leave it to them. We cannot go trawling and finding all of the genomes that exist in different databases. So it has to be submitted to the INSDC for us to pick it up.
I'll take the Bar Usman's question of how can I submit my proteomics data to UniProt? So we don't tend to take proteomics data directly unless you've got a novel sequence of a peptide. So that isn't a peptide that currently isn't known in our proteomics data set. If you've sequenced that and you've pulled that out of your mass spec, then you can submit that with a tool to the UniProt website called Spin, and that's you can find that through our normal web URL. But typically, if you've got a big proteomics data set and you've got a large amount of data, you submit to PRIDE, which is another EMBL-EBI resource. And everything that's submitted to PRIDE will get filtered into UniProt anyway. And it's a much more efficient way of doing things with regards to submitting large data sets as well.
Check out the other webinars we have in this series on UniProt. Also wanted to mention that we have a release every 8 weeks. And this is the link to our online training. And please get in touch at help@uniprot.org.
So we have a question on how to download specific proteins. So if you've got a protein of interest or proteins popped up in your research that you'd like to download all the information for, just go onto the UniProt webpage and in the search bar at the top, search the name of your protein or indeed a gene name as well, and select your protein from the list. It will detect synonyms of names as well. So if we've got a gene synonym in, so put that in there and it'll bring up the correct gene name for that. And you can, if you click on your protein of interest in the list there, you'll get the protein entry page and all the information that UniProt currently has for that protein. And on that page, there is a download button to download all the information. And that includes feature information, function, interaction information. Essentially everything you see on that web page you can download. If you're interested in the peptide sequence of that protein, you can also, in the same download button, there is an option to download faster canonical sequence. So that is the canonical amino acid sequence that we have for that protein in the database. And you can also download isoform information as well if you're going through the API, which is explained in the proteomics webinar as well. But essentially, when you're downloading information like that, you've got the option to download specific information. If you just want to pull feature information out or just interaction data, you can do that, or you can just download the entire protein information. Another way of doing it is if you've got 5 or 6 proteins you're interested in, if you search them one by one in the search bar, and there is an add to basket function on the website, much like when you do your shopping, you can add that protein to basket, search for another one and add another protein to basket. And then when you're done and you've got all your proteins of interest, you can go to your basket and select what sort of information you want to download from all them entries as well.
So we have a question from Ritwik about how to find the proteome of a particular tissue or organ. And that's a very good question, but unfortunately we don't have that yet. Hopefully we will have that level of information in the future. But at the moment, it's a proteome is the set of proteins expressed by an organism.